It's an honor to be with you here today. I want to thank the organizers for the opportunity and the panelists for having us here today. I'm excited to tell you a little bit about Aquea Health after being in stealth mode for quite some time, and to introduce today our novel MIGS platform, which is a novel, physiologically relevant architecture for MIGS that we think has the potential for best-in-class performance with respect to efficacy, safety, and ease of delivery. And I'm excited to talk to you today a little bit about how this specific architecture matches the needs for performance within Schlemm's canal specifically and in the hands of the surgeon. Our team, we're very proud of, our co-founder, Andy Doraiswamy, deep experience in ophthalmology, inventor on the Invista lens, ran Santen's US operations, Ocular, now Aquea Medical, Mark Packer, our chief medical officer, who needs no introduction, I'm sure, and has been just a wonderful team member with us, and Dan Hamilton, who heads up our Clin Reg and has been in that space as a senior executive literally for decades. And then we're thrilled to have a terrific board and supportive investors that have financed us as well. Cutting straight to the technology, this is an image of our device. It is engineered to provide patency within Schlemm's canal while providing access to the collector channels within it. I'll show you a little bit more as we get through some more images. And a delivery mechanism that is designed to be intuitive and easy to use in the hands of the glaucoma surgeon. This architecture and specific design enables performance across a variety of factors, one of which is highlighted here, which is to provide patency within Schlemm's canal. And here you can see a cross-section on the, your right, of our device in cadaver eye. And you can see how much patency, how much space within Schlemm's canal is created for the amount of material that is present at that particular plane. And on the left-hand side, the tightly wound portion of the device that provides bypass flow into Schlemm's canal. And to bring it together, let's go to a video here. This is one of our very first patients in our feasibility study that we conducted. And as we roll here, you can see how naturally this takes to Schlemm's canal, the blunt end of the device, entering and proceeding benignly within the channel, the wide open architecture of the stent providing space and scaffolding is what I'm looking for within Schlemm's canal, and then the tightly wound portion that sets up as a port from the anterior chamber and into the canal. Our first human study was very successful. This was designed to show that the device could work and to provide safety, and it certainly demonstrated that. These patients weren't washed out, so we weren't looking for an efficacy signal, but we were very pleased with the efficacy that we were able to show anecdotally with respect to lower IOPs in patients coming off meds. The eyes have been very quiet out to 12 months and beyond, no occlusion, no migration, no inflammation. Based on that, we're conducting two phase 2 studies that are presently enrolling, and we look forward to reporting out on that data when it comes later this year. Importantly, I wanted to highlight our intellectual property position, our fundamental patent issued last year. I'm very pleased with the protection and freedom that provides us. So in summary, a next-generation MIGS solution that we think provides intuitive method of action within a known anatomy that is built for really efficacy, ease of use, and safety Pleased that our team is executing as well and as leanly as it is, and compelling feasibility data to come. On the left-hand side, this is a nice image of the device out at 12 months in a patient. You can see how it sets up within the anatomy and how much space is created in a place where you're looking for unrestricted flow. I'll pause there.
Thank you, everyone, for being here today. It's a great honor to join you, and thank you to the program chairs and wonderful moderators for inviting us to participate. My name is Ruijing Zhang. I am the founder and CEO of Avisi Technologies, here to give an exciting update on VisiPlate aqueous shunt. At Avisi, we're developing nanotechnology-enabled treatments for glaucoma. We're venture-backed, based in Redwood City, California, and the technology comes from the University of Pennsylvania. Our first product is pictured here, VisiPlate, an ultra-thin multi-channel device. We've shown safety and effectiveness in lowering intraocular pressure in early OUS human clinical trials, which will be presented upon for AGS later this year, or later this month. We believe that innovative materials are the key to improve glaucoma surgery. And we say this because gold standard trabeculectomy continues to involve risks like fibrosis, hypotony, infections, and blood complications. Meanwhile, the devices we have today continue to clog, erode, migrate, or require frequent revision due to an inherent mismatch between the materials used and the tissue. Ultimately, all current surgical interventions may fail from occlusion and scarring. So this is where VisiPlate comes in. Our device has a patient-centric design for longevity, comfort, and patient aesthetics. We use non-fibrotic materials, we have a multi-channel design to create outflow redundancy, and we have a surface area to maintain the drainage zone over time. The device is 5 microns in thickness, so you can see it balanced on the fingertip, or maybe you can't, because that's about 20 times thinner than a human hair. And it's our metamaterial technology that ensures handleability despite this scale. VisiPlate drains to the subconjunctival space, which is a space we're all very familiar with. And the idea is to wick aqueous in a slow and controlled way through a network of microchannels. The surface area of our device is able to maintain that drainage zone well over time, enabling, again, a very low and diffuse drainage zone that preserves comfort and aesthetics for our patients. So in keeping with the Maverick, or M theme of this panel, I'd like to introduce the 3 M's of VisiPlate: our materials, our mechanism, and our surgical methods. Our materials are ultra-thin and non-fibrotic, and the clinical advantage to this means less anti-fibrotic use and more patient comfort and aesthetics. The metamaterial is a nanometer scale alumina core coated in perylene C. These are highly biocompatible materials that are also isotropic, flexible, and conformal to the globe. Our device is also asymmetric. We have a top side filled with hundreds of microchannels facing the conjunctiva for drainage, and we have a bottom side with wells for tissue ingrowth as well as future applications in drug delivery. Here we're showing a comparison of chronic New Zealand white rabbit data where we implanted VisiPlate for 6 months with no mitomycin. And you can see that we were able to maintain open drainage channels and no fibrotic capsule formation, versus other studies also done in New Zealand white rabbits without mitomycin, for example, silicone tube shunts. At just 2 months of implantation, you already see a capsule forming of 450 microns. And this is to underscore, again, the truly unique nature of our material. On the mechanism, VisiPlate is the only device with multi-channel redundant outflow. And the clinical advantage of this is to avoid failure from clogging. While other single lumen devices or 1, 2, 3 lumen devices may clog with debris or scarring, as you can see from this CFD model, VisiPlate is not prone to clogging with these surface occlusions. In fact, our device is self-correcting. The flow actually accelerates around surface occlusions like this. And on the methods, simple, straightforward. You don't need any gonioscopy, for example, intraoperatively or postoperatively. You don't need to change the way you sit or the way your scope is oriented. There's no special tools required, and it's a quick learning curve. In terms of the procedure itself, we make a peritomy at the limbus. You make a scleral tunnel a couple of millimeters back with a keratome blade. You insert our device with forceps of your choice fixate it, and close the subconjunctival pocket. Here you can see the relaxed atmosphere in our OR and a beautifully healed eye 9 months after implantation. So in summary, VisiPlate uses non-fibrotic materials to preserve the drainage zone over time. We have a multi-channel outflow to ensure patency and redundancy. We have an ultra-thin profile for patient comfort and aesthetics. And we have an easy surgery to fit into the existing workflow. I'd like to give a quick shout out to our team. We have wonderful experts supporting this company, and we wouldn't be here without them, so thank you all. Coming up next, Dr. Edie Miller-Ellis will present VisiPlate clinical data from the VITA clinical trial at AGS later this month. We also aim to launch Sapphire, our US pivotal trial. And for more updates, please do follow us on LinkedIn or check out our website for more pipeline information. Thank you so much.
Thank you. Very exciting.
Guys, we're missing big pictures in glaucoma all over the place. So in Japan, 90% of glaucoma patients never had a pressure that was higher than 21. In the United States, 30% of patients that have glaucoma never had a pressure that's higher than 21. And in the clinical paradigm, it's really easy to take an IOP that's 25 and get it down to 18. I've got about 20 different things that I can use every day that can do that. But it's pretty hard and it's risky to take a pressure from 18 to 12. It's hard to lower eye pressure at night. Most therapies don't work that well. It's hard to do it in addition to whatever therapies they've had before. So normal tension glaucoma is hard to treat and it's bad. 50% of treated eyes in normal tension glaucoma can achieve a 30% IOP lowering. 34% of those NTG patients still show progression. 9.9% end up going blind in one eye and 1.5% go blind in both eyes, yet the treatment is still to lower the eye pressure. And if you can achieve a 40% reduction in IOP, you can decrease the progression rate by 5.7-fold. Recently, and came out of this meeting 4 years ago, there was a combined AGS-ASCRS paper about the unmet needs that remain in glaucoma. They talked about the importance of lowering IOP 24 hours a day and the need for non-invasive therapeutics to lower IOP, especially in those patients who don't adequately respond to therapy or whose pressure is already in the normal range. And so here's our answer to that. It's the Physics Ocular Pressure Adjusting Pump. It has 2 pumps in the pump mechanism that allows us to draw a negative pressure vacuum over each eye independently. It's the first non-surgical, non-drug way to lower IOP, and it was approved by the FDA in June of this year, of 2024. Prior to that, we went to FDA panel and we won 13-0 at FDA panel, which was the highest level of scrutiny that the FDA provides. It lowers IOP in every eye, every single time. It does it safely, and it can do it on top of medications, and it can do it regardless of the baseline IOP. It only works while it's worn, and that's at night while we sleep when eye pressure normally goes up and blood pressure goes down. We have the ability to monitor usage to address compliance issues, and the FDA label is that it's indicated for the reduction of IOP in adult patients with open-angle glaucoma whose pressure is less than or equal to 21 at the time of treatment, that have either tried or are currently on glaucoma therapies. How does this work? It's simple. It really is just physics. We are pressurized by the weight of the atmosphere on our entire body. All we do is release a little bit of that weight over the eye. So imagine if I pushed on your eye pressure has to go up, and when I stop pushing on your eye pressure has to go down. We're just not letting the atmospheric pressure push so hard on the eye. Now, some people thought that this doesn't actually lower IOP, so I'm going to remove that doubt right now. I'm going to show you what you're going to see. You're going to see a manometer inserted into a patient's eye that's having cataract surgery, and you're going to see that we can dial in the eye pressure to whatever we want it to be. So these are 17 patients that were having cataract surgery. This is a laboratory version of the pump. We're dialing in a vacuum of -10. You can see that we're dialing that in and we're measuring it, and you can see immediately in the eye, it goes from a baseline of about 14 millimeters of mercury down to 9 millimeters of mercury immediately. If you could fast forward to the 1 minute and 25-second mark on the video, now you'll see that we're going to dial in -20. Watch how much the IOP lowers. There's the -20 of vacuum. The eye pressure responds almost immediately and goes from 15 millimeters of mercury down to 7 millimeters of mercury. It is really hard to take a pressure from 15 millimeters of mercury down to 7 millimeters of mercury. Here's the setup in the operating room. We're grateful for patients that are willing to consent and be part of these kind of trials. So we've done 2 multi-center randomized controlled trials, one with 128 eyes, one with 182 eyes, one in open-angle glaucoma, one in normal tension glaucoma. The one on the right is the one that we used for FDA approval. We found very similar results in both studies. An IOP lowering of 34% from 19.4 to 12.9. And in the normal tension group, pressure measured at night in sleep labs during COVID, not that easy, that's why we called it the Hercules trial, from 20.2 down to 12.2, 8 millimeters of mercury of IOP lowering of 39% lowering. This is the consistency through the beginning of the trial to the end of the trial, between 37% and 39%, again from 20.2 down to 12.2 millimeters of mercury. This is something that I'm really proud of. We've done 6 different randomized controlled trials, 18 peer-reviewed papers, over 650 eyes studied, and when we look at it physiologically, we show an improvement in blood flow. We've shown a decreased stress strain on the optic nerve head. We show an increase in the percent area perfused and a decreased capillary dropout. And so as we think about those patients that are the hardest to treat, those that are getting worse with a pressure of less than 21, facing significant surgery that carries big risk, we believe that the ocular pressure adjusting pump can help fit this category.
Well, thank you, John.
Good morning, and thank you for the organizers for this opportunity to present in this forum. I practice as a glaucoma surgeon in Melbourne, both as in public and private practice. I'm involved in a number of innovation projects. One of them is called the Melbourne Rapid Fields, which is an AI-enabled online perimeter for home and clinic-based use. And also I'm involved in a number of surgical innovations, and today I'll be talking to you about 2 tube shunt designs that we have PCT patent accepted. So as a glaucoma surgeon, the features that I particularly look for as ideal features are having good long-term and short-term pressure lowering, avoids early hypotony, ease of implantation, minimal tissue dissection, and low complication rates. And what my thoughts are is how to improve the existing non-valve tube devices to achieve some of these ideals. Why non-valve tube shunt devices? It's because they have several advantages. They have been around for a long time. There's excellent history of efficacy as evidenced by the TVT study. They are less affected by conjunctival scarring because of its position behind the equator. There's less risk of bleb leak, less manipulation and less follow-up, and mitomycin C is often not required. And for non-valve tube shunts, IOP can be further lowered in the postoperative period with complete tube opening. But they do have disadvantages, and they are difficulty in achieving early pressure lowering without hypotony. They require larger conjunctival dissection, and the operation can be a bit more complicated. Of course, recently it's been shown that you can achieve early pressure lowering without hypotony if you have the optimal combination of tube internal diameter and intraluminal suture diameter, but it does require that there you have a change in the tube diameter, which can be a manufacturing challenge, and also it can be a risk of occlusion with smaller internal diameters. So one of our first tube shunt design is to re-envisage the venting slits or holes that glaucoma surgeons that are currently using in non-valve devices, and making it a pre-manufactured feature. The benefit of this is that it can seamlessly integrate into any non-valve devices, and the venting slits or holes can be designed using microfluidic engineering to ensure that the early pressure lowering achieves a physiological level. And this is accepted as a PCT patent. The advantage of pre-manufactured venting slits or holes is that it leverages the well-established method of pressure lowering and is easily understood by glaucoma surgeons around the world. And there is also an option for the surgeon to either use or not use these pre-manufactured venting slits or holes by the strategic placement of occlusive ties. How about making the tube shunt more minimally invasive? In our second design, we envisage a modular design for the tube shunt, where our standard tube shunt can be divided into a plate component and a tube component, and you can have an injector system for both components. And when the two components are placed inside you, it can be connected using a connecting mechanism to achieve a functioning unit. To make the plate more easily stable and more efficiently stable, we also envisage sutureless methods where a latching system on the plate can be used to latch onto a scleral tunnel without suturing. So in terms of insertion technique, it can be achieved with a smaller conjunctival incision, and the two components can be inserted separately inside you and then connected into a functional unit. And a single conjunctival suture can be used to close the conjunctiva. So in summary, we want to change the glaucoma tube shunt surgery so that it can combine the safety and speed of mixed procedures with the efficacy of tube shunt device. And we are now seeking collaboration with manufacturing partners to make these innovations into reality. Thank you.
Hello, and thank you so much to the Glaucoma Research Foundation for inviting us to be here today. My name is Rachel Chapman. I'm a third-year medical student at University of Central Florida College of Medicine and an aspiring glaucoma specialist. And we're going to rewind a little bit here first to think about patients before they're entering our clinics with glaucoma. So, if I can make this. Oh, there we go. So a quick introduction. I became interested in ophthalmology as a scribe in the retina and cornea clinics at the University of Virginia. And when I got to medical school, I thought I'd maybe missed out on glaucoma a little bit. So seeking out research, I had the absolute privilege of working with Dr. Arthur Sitt at the Mayo Clinic in Rochester and presenting our research at ARVO. But when I came back to medical school, I couldn't stop thinking about how we catch glaucoma. And I knew we had such great tools in the ophthalmology clinics with Humphrey visual fields and RNFLs, but what about the patients who aren't going to see ophthalmologists? I know the AAO recommends a comprehensive eye exam at 40, but most of the patients I know, if they don't have visual symptoms, aren't seeking out that kind of care. And so I started to think about what type of screening and risk assessment was available in the primary care setting to understand and educate patients before they became symptomatic with glaucoma. And to be honest, there wasn't a whole lot out there or a whole lot of research on it, which was surprising to me because there's been great epidemiological research done on glaucoma, and we have great new technologies that could make risk assessment a lot more accessible. So, using my ideas and trying to combine what technologies were out there, my co-founder and I competed in the Florida Blue Health Innovations Challenge and won this past April, which gave us the seed funding to start EyeSentry. So, like I said, we're still in the development phase, but what we're designing here is a provider-facing tool that will help identify high-risk patients in the primary care setting who haven't been connected with an optometrist or ophthalmologist. And the way that it works is by an easy guided system for the physician or physician extender to use that incorporates tonometry and new handheld fundus imaging to measure cup to disc ratio. So we're incorporating a more comprehensive screening tool. Probably most importantly, we can use this for prospective analysis to get a better understanding of the value of risk assessment and screening before that comprehensive eye exam at 40, which we so lack because year after year, the USPSTF has not been able to determine a guideline based on insufficient evidence. So, where are we now? We're currently on track to complete our software development this year, and in the meantime, we're working on building partnerships in industry to help with EMR compatibility, coordinate with tonometry and imaging companies, as well as talk to our friends in public health and health insurance to understand how we could implement EyeSentry in clinics like the Florida Blue Sanitas clinics, as well as federally qualified health centers, and talk about what reimbursement will look like for using this screening system for our primary care providers. Now, we hope to launch by my graduation from medical school in 2026 with a prospective analysis. So, let's think about what this would actually look like to use in the clinic. So, the physician opens their EMR, as per usual, selects health maintenance, and this populates which screenings are available for the physician to conduct, and EyeSentry will populate there. Now, we think about introducing this maybe between the ages of 35 and 40 to further initiate people getting that comprehensive screening eye exam at 40. Once the physician selects that, it'll redirect to the application where there'll first be a pre-screening to determine whether the patient's already established with an eye care provider. If it's no to either question, they can initiate the screening, and it'll auto-populate from the EMR demographic factors and then guide them through questions assessing first-degree family history, myopia, as well as any past history of topical or systemic steroid use, and then provide not only written but also video guidance for using a handheld tonometer, and similarly for our new non-mydriatic handheld fundus imaging cameras to measure cup to disc ratio, something that hasn't been incorporated in the past. So once that's complete, EyeSentry will provide a risk assessment category, high, intermediate, or low, based on the number of risk factors that were accounted for. And this will continue to improve as we gather more data and can use machine learning, different vector methods to better the categorization. It'll also provide an idea of how strong the evidence was. You know, did the provider have access to a tonometer or a handheld imaging camera so that they could include that data or not? And then it provides resources for the provider to have a discussion with the patient and undergo that shared decision-making that's so important. So providing information on which risk factors contributed, as well as information on what timeline, as well as which resources it would make sense to the physician to send the patient to, including, you know, local eye care providers, as well as federally qualified health centers with vision care resources, and then printable patient resources in English and Spanish to help them further understand their condition and think. Now, a recent estimate projected 67 million cases of undetected primary open-angle glaucoma by 2040 worldwide. And we're really interested in significantly cutting down that number. Thank you so much to the Glaucoma Research Foundation for including us here today. If you're interested in EyeSentry, please come and talk to me or my co-founder Hannah, who are here. Further, our contact information is available at these QR codes. Thank you so much.
Okay, good morning, everyone.
First of all, I'd like to thank you to be able to see what we're going to present today in terms of a transformation, how we measure in IOP. I'll do this. Okay, one of the elements that really came to us a while ago is that when we look at actually the data that we talk about in terms of IOP is a number. But when you think about it, is we're living on the side 90% of the information that we need. And that's where I think we started to focusing in terms of not looking at one numbers, but looking at a profile. So when you think about that aspect, now you have to find out how you're going to get this profile, how you're going to get this large amount of data that you're going to need to really understand how the IOP is affected by therapy. So the key element that we have is how do we position a sensor in the eye to really capture that information. And first of all of us are focused on the front of the eye, but we all know that the pressure we want is a pressure seen by the optic nerve head. So that means we need to position the sensor in the back of the eye. So the key element, if you go into this position, is how you can implant the device with minimum risk for the retina or the lens. So in fact, you see the name of the company is injecting a sensor. So we're implanting the sensor in a pars plana, and we self-anchoring the device on the sclera. So the procedure is a 2 millimeter self-healing incision. And that way, the sensor is now able to capture this pressure all the time. We can go down to 10 millisecond interval to capture the pressure. But now what we have is a continuous access to IOP pressure. We don't rely on the patient to acquire the data. And what we additionally added to this is the battery. And embedding a battery in the sensor allows us to have continuous monitoring, including at night, which is an area of interest. And the key element now that we're able to connect the sensor through a pair of glasses to the cloud, we have the ability both for the patient and the physician to see how the profile evolves over time. So the key element, why this information is necessary, we all know we have to use now AI, and to train an AI model, we need a large amount of data. And this data has to be unbiased, unfiltered, unaffected by any element like how you calibrate the device or how you use it. So we need to take as much of the patient out of the equation. So here, you can see the device. It's a plug. and the plug is delivered into a couple of minutes procedures. You can see the device in the pars plana. Why is the pars plana so interesting? We today doing millions of injections for anti-VEGF. With anti-VEGF, sorry. So the key element, you can see the sensor locked in position. As a side note, we have already a handful of patients with the device currently in Santiago, Chile. And so the key element for us is now we are able to collect all that information. And the side note is the FDA and the NIH have looked at this very carefully. So it's not just science fiction, it's real science that we had people looking it over. So now what we have a common tool. Everybody have a cell phone available. So from the patient point of view, now you can see the immediate effect of the therapy, either today, tomorrow, in a week. So this follow-up of that pressure will definitely reduce the anxiety, and you will understand, for example, how to be more compliant, how to follow his drug regimen based on the information available. Again, this data is all available on the cloud. From the clinician point of view, now you are, in fact, your therapy is shaping the curve. So we're not looking at a data point. You have ocular hypertension, you bring it down. You can see here the way we present the data is really kind of a way to look at the IOP wobbling during the day, at night. And this visibility is very synthetic. You don't need to look at data point. You see the pictures, you assess where the excursions are, and you can adjust the therapy, and you don't need to wait 6 months to figure out what to do. So the simplicity of this approach is to really compress the information to something clinically actionable. As you see at the bottom now, what you're doing here is you can start to open up the domain of multimodality. You can connect to OCT, you can connect to visual field, and those are the domain where quality information is extremely critical. And all the AI tools that we see now developing will absorb that information very easily. So now we're into human. So that has been a long road to get to this point. So we're doing that in Santiago, Chile, with Dr. Mura. The first element that we have already, and it's the tip of the iceberg, we've already established safety of the device into animals. We currently have about 15 rabbits with basically no safety issue. But the key element, once we're in human, now we have to understand how to deliver the device, what are the conditions of the tissues. So we have to really understand the biomechanics of the eye. So here, sensor delivery. This is a procedure where the sensor is delivered in position in a very controlled fashion. You can see over the slide, we start positioning the device at 3.5 millimeter. We push the sensor in position. We have shift the conjunctiva by 1 millimeter over the cornea, so that way when we release the injector, the cornea is not, the conjunctiva is not over the device. Now you can see 4 months later, the device basically is waist locked in position, and the patient a few weeks later doesn't even know he has a sensor. So here we are inspecting the device, we're using UBM, all the tools that we have available in an OR are available, but the key element is that procedures will allow us to go initially in OR all the way up to in-office procedure. Why? It's because there's no suturing, it's a self-healing incision, and the patient after that is connected to the cloud. In terms of data, our goal is not to focus on single data point. Statistically, we can measure the equivalence or limit of agreement with GAT or any other technique. Our goal is to measure a profile. So now, where we go from there, we are finishing up a group of about 20 patients. We are informing our design to go to the final product. But because of the importance of doing this into a supply chain, we're anticipating, and you can see here the building that we are about to build in the next 6 to 9 months is going to be the recipient of our supply chain, producing about 10,000 devices per wafer. And that will allow us to go into large quantity and then provide this device to a lot of people. So I think this is a paradigm shift, and I'm sure you have a lot of questions down the line. Thank you.
Thank you.
Thank you. Good morning, everyone. First of all, I want to thank the Glaucoma Research Foundation for this invitation. We're very honored and we're very thankful to be here. This is indeed a very important day for us at Blue Ophthalmic. My name is Carlos Herrera. I'm the Chief Medical Officer, and I'm here to introduce the AquaLumen. It's a new surgical device that is commercially available in the US and that we hope will help bring back filtration surgeries into the mainstream. We really care about this daily trade-off that physicians and patients are experiencing related to safety and efficacy. On the one hand, as we have heard multiple times today, we have MIGS with a great safety profile, but with a limited ability to reduce IOP. And in the other hand, we have trabeculectomy, which has a much better capacity to reduce IOP, but comes at the cost of the potential risk for significant adverse events. With that in mind, this is what I want to do today. We're presenting the AquaLumen. I'm going to share with you how the AquaLumen is changing that equation. So the AquaLumen is an implant-free ab externo device that will allow for this minimal invasive filtration surgery. It is precision engineered to create a standard communication pathway between the anterior chamber and the subconjunctival space. And this is the mechanism that is similar to that of a trabeculectomy. What I'm going to show here in this video, this is actually an animation of the procedure. We see the device here with an ergonomic handle, a bent ankle that allows for easy entry into the anterior chamber, and that tip at a notch that will actually allow to remove the tissue. So as the eye is being prepared, similar to other filtration surgeries with mitomycin, here's the key point of the surgical procedure. We see how the incision goes into the anterior chamber, and in this close-up, you see that entry into the anterior chamber, that notch actually takes part of that scleral tissue, When you bring that scleral tissue out, you create that lumen that results in this low-lying diffuse bleb. Here is the procedure now being done on an actual patient. This is Dr. Rodolfo Perez-Grossman, the inventor of the procedure. In this case, he's using a Vera hook for traction. Some other physicians have used suture traction for that, which allows for that physician presence. We see the device going into the anterior chamber. You see the tip at this point going in, and in that motion of the excision, when you come out, you bring in a pus part of the tissue, and then you see right there the formation of the bleb. You remove the AquaLumen, and then you're done with the procedure, no sutures or anything like that. So minimal conjunctival trauma there. Sharing with you now some of initial clinical experience. These were 33 eyes in 27 patients. One of the things that we want to highlight is that we've seen higher IOP reduction that is more pronounced in patients who have a higher baseline. So this is compatible or comparable to the more aggressive filtration surgeries. The same data now we're looking at a percentage reduction. Again, higher baselines, higher percentage reduction. And one thing that we wanted to highlight here is that those patients have not required the use of glaucoma concomitant medications during the post-op period. Now we're looking at a group that has a lower baseline, lower than 21. We also saw the reduction of the IOP, and we also want to highlight that there were no adverse events, no serious adverse events reported, and those adverse events that we had, the non-serious ones, were consistent with similar procedures. Now we're going to take a look at some images. Here are some blebs at different time points, 2 weeks, 2 months. Here's what that bleb looks on OCT. Here's another OCT, but at 4 months. And now we're seeing a patient with that bleb at 8 months post-procedure, and this is the corresponding UVM at that time period. And I'm just going to highlight here that you're actually able to see the lumen that we talk about. We're a US-based company with headquarters in Miami. Here are the 3 co-founders of the company. There is one person that I really want to highlight in particular. This is Dr. Rodolfo Perez-Grossman. He's, of course, here in the audience. In addition to being the inventor of the technique and the device, he's also a former Schaefer Fellow. So that's a great testament. It is a fantastic story of the impact that the Glaucoma Research Foundation through their fellowship program has had. So now he's back, a few years later, give or take, with an invention and a company. So that's just fantastic. We're backed up by a great group of medical advisors. They've been wonderful. We're extremely thankful to them. They have certainly helped shape the path that we're taking on as a company. With that in mind, I want to highlight a few things. So again, we're really closing that gap between the safety and efficacy profile of the filtration surgeries. We have very promising clinical data for which we're very excited, and then we're commercially available in the US and obviously happy to entertain partnerships to expand our commercial efforts. Finally, I'm going to leave you with some thoughts from our advisors who have had the opportunity to do the procedure, and you can hear from them directly.
Is the incredible post-operative outcomes that we've seen today in the patients that had surgery yesterday and weeks ago. These eyes are extremely quiet. The patients are very happy. There's very little inflammation. They have the most beautiful, low, diffuse filtration blebs that I have seen.
It is something that can be done more rapidly, and more importantly, it has a greater safety profile so that there's probably going to be less risk of hypotony, perhaps infection as well, and it's going to have closer to a safety profile of the existing MIGs that are out there.
I do think this type of procedure, a stentless procedure creating a bleb that's predictable, that's reproducible, gives us an opportunity in those kind of patients where they're not early enough like a conventional MIGs, and we don't want to do a traditional trab or tube. As that large patient population, I think this could really fit in very nicely.
It's a quick and easy procedure, and one that I think should be adopted by most glaucoma specialists, especially when they find out how easy it really is to use.
Thank you.
Thank you so much, and huge thanks to the organizers for the opportunity to present here. This is a really amazing session, and I'm loving it. So I'm Ross Ethier. I'm a co-founder of an early stage company, Serene Ophthalmic. My day job is to be a professor at Georgia Tech and Emory University in Atlanta. So what is Serene? We are an early stage company developing a device that uses a non-surgical approach to deliver an inert hydrogel spacer into the suprachoroidal space. And I'll show you data that indicates this achieves significant long-term IOP lowering. We expect this would be adjunctive to beta blockers and therapies that target the meshwork. Importantly, it can be repeated, so you can re-inject that hydrogel with high efficacy, and it has an excellent safety profile both in rabbits and non-human primates. So a little bit about Serene. The co-founders are myself with a background in glaucoma, my colleague Mark Prausnitz, a biomaterials expert and a serial entrepreneur, and Mike Garanzini, an experienced executive in the ophthalmic space. We have an all-star team of medical advisors, as you can see from this slide here. So what about the technology? As I mentioned, we inject this inert hydrogel into the suprachoroidal space that physically expands that space and facilitates drainage through the unconventional pathway, lowering intraocular pressure. What are the advantages? Well, there's no surgery. It's an injection. So it can be done as a rapid in-office procedure. It's easily performed after some brief training. And I'll re-emphasize, there's no drugs here. It's a soft, inert hydrogel that's delivered. And that word soft is important. We're not putting a hard material into the suprachoroidal space, so that reduces concerns about erosion and significant fibrosis. So we use a microneedle with a length that allows you to safely deliver the hydrogel into the suprachoroidal space. There's a single injection that's done at a single location, about 2 millimeters posterior to the limbus. The hydrogel is delivered, forms a small bubble which resides in situ within the suprachoroidal space. Now we have a number of instantiations of this hydrogel. I'll refer to them as the first through the fourth generation. And let me share with you some efficacy data. So this is intraocular pressure data. It's the difference between pressure at a given time point and the baseline pressure. This is in normotensive rabbit eyes with our second generation formulation. The gray symbols at the top, that's a sham injection, so no pressure change as you would expect. But if you look at the black symbols, you'll see after injection you get about 5 millimeters of mercury pressure decrease, which is about 35% in these animals. And then slowly over time, that pressure creeps back up towards baseline. You net about 4 months of pressure lowering. We did these same type of experiments in cynomolgus normotensive eyes. You can see a similar behavior of the intraocular pressure there qualitatively. And we also measured the thickness of that hydrogel bubble within the eye in vivo. And you can see in synchrony with the pressure creeping back up towards baseline, that hydrogel slowly diminishes in size. And you see a very similar set of data if you look at hypertensive eyes. Here's some ultrasound biomicroscopy data. Just on the left side, we've got the sham injected eye. This is at the site of injection. Looking over time, there's nothing remarkable. On the right side, you'll see the hydrogel injected eye. And if you notice, just after injection, there's that orange outline, essentially a little bubble of hydrogel just posterior to the iris. And that decreases in thickness over time. So now this loss of hydrogel over time and the associated drifting of the pressure back towards baseline is not optimal from a clinical viewpoint. So we went on to develop a third generation formulation. This is again in normotensive rabbit eyes. Here's some of our second generation data just to remind you what that looked like, but please focus on the colored curves here. We're getting between 300 and 450 days of pressure lowering after a single injection. And that's about 25 to 30% pressure lowering. And we've got a fourth generation formulation now that we're up to 700 days plus. So basically 2 years of pressure lowering after a single injection. So there's some other data that I won't go into the details of, but we have an excellent safety profile with a number of outcome measures. I mentioned that repeated injection is efficacious. So if the pressure drifts up towards baseline, you can re-inject. Basically, you get the same bang for your buck the second time around. We have data that shows that it reduces diurnal pressure fluctuations, and we also have good mechanistic data showing that the mode of action is by improving unconventional outflow. So just to try to summarize, we're really excited about this technology. We have a good IP position around use and composition. I've shared some of our preclinical data around efficacy and safety. We have a long duration of IOP reduction, and of course facilitating unconventional outflow is an accepted mechanism of action. Just looking forward, our regulatory consultant indicates that he expects a favorable regulatory pathway as a device rather than a drug. It's a simple, quick in-office delivery. We have a strong team, and our funding to date is non-dilutive. With that, I thank you so much for listening and welcome questions.